Good afternoon, students. Welcome to my PP class. Hope all of you are well and following the rules, whatever given by the government regarding the coronavirus, and doing full justice with your time. Today, I will discuss another important disease, that is powdery mildew. First of all, I will discuss the powdery mildew disease as a whole, and thereafter I will discuss the powdery mildew of cucurbits in detail. So the powdery mildew disease, from its name, it speaks itself. How? Because somehow this name indicating that it is a disease where something powdery-like substances is associated. That means anything powdery-like growth or powdery-like materials related with this disease. So it's a very severe disease of the crop. Almost every type of crops affected by this powdery mildew. And this disease, especially it prefer little bit lower temperature and dry weather. So that is the main reason that this disease you will find every part of the world, and it causes almost every crops. Of course, the gramineous plants are little bit less infected by this pathogen. So this powdery mildew disease, it's a very important disease, and it infected near about 700 species of the host throughout the world. And if you see the damage, it's near about 40 to 60% reduction in grain yield. This is in case of powdery mildew of barley in Australia. The damage level also vary from place to place, from season to season, but more or less this powdery mildew, it's a very problematic disease for everywhere. This is a disease, it is very easy to identify because due to the presence of the whitish powdery growth on the substance, powdery growth materials or substances on the surface of the leaf, you can identify the plants quite a far distance in field. So it's a very easily identify and recognize disease, and most all the above ground part are infected by this disease. So the powdery mildew pathogen rarely kills the plants, okay, but it interfere with the photosynthesis, it interfere with the respiration, transpiration, and it interfere with the growth of the plants. That is why there is a drastic reduction in yield. Sometimes the powdery mildew disease, it kills the plant also in the later season. It is going to be a problematic in greenhouse plants where the cool and dry weather is very much common, you see. So it's a obligate pathogens. Obligate pathogens means it is very much growing on the living host only. It can't survive as a saprophytic nature. Saprophytic means, that means it can't survive on the deep plants materials. But nowadays we can culture the pathogen Bloomeria graminearis, which is infected in the oat. This pathogen now we can culture in the artificial medium. So there are various type of genera which causing powdery mildew disease. This is very important because number of genera. You see, the Plumeris graminis. It names from the gramineous. You can identify that it is causing the gramineous plants. So it infect the gramineous plants. But I already told you that the infection is gramineous plants is little bit less in compared to the vegetables and other dicotyledon crops. Irisepi sicurisirum. This is another species causing disease in cucurbits. Irisepi polygony, where it's infecting the legume crops. So this recipe is a very common species, common genus for our country on India. Then there is the Libellula. It causes the powdery mildew in tomato, very common in USA. Mycosphaera, this is another species causing powdery mildew in shade trees, in very plantation trees, woody plants. Oidium, this is another genus which is causing powdery mildew in tomato. Phyllectenia, causing powdery mildew of shade and forest trees. Podospora, causing powdery mildew upon pear. Nowadays it has been clearly indicated that podospora causing in cucurbit also, and this podospora is the main causes of cucurbits. Okay, Sparotheca, it's another important genera causing powdery mildew in strawberry and so many, even in rose powdery mildew nowadays is becoming very popular important disease in USA, in Israel, in European countries where they are cultivating the rose in a commercial scale. Sparotheca fuligina, these are the names. Sometimes you can say it's a synonym of the same pathogen, or sometimes like that. Uncinnula is very important. It's causing the grape powdery mildew. Then Uncinnulella, that is also another species powdery mildew, horse chestnut. This is not common in our country, but these are the pathogens. You see, 1, 2, 3, 4. So many pathogens are causing the powdery mildew disease. You have to know very critically, or you have to know very carefully that these are the pathogens causing powdery mildew disease. And these powdery mildew pathogens, these are generally differentiate on the basis of formation of the number of ascus in Clistothecium. These are the Clistothecium, you see, these are the Clistothecium. And in Clistothecium, what happened? There are some genus which forming only one ascus, whereas some forming many ascus. Okay, you see, there are many ascus. So by this way, this is one character. Another character is the presence of these Clistothecium appendages. Here is the appendages. This is the style, different fork type. Here you see there is a bulbous base. Here you see the hook type. So based on these type of various type of mycelial appendages on the Irisaceae, on the Clistothecium, the pathogen has differentiated. Now the Clistothecium we are also called as a Chasmothecium. It is a new name of Clistothecium. Earlier we are calling it as a Clistothecea. Now it is called Chasmothecea. There is hardly any difference. These are the same, but name has been changed. So you see, these are the microscopic. In microscope, you will see this is the oncylal equator. Okay, clearly visible. There is a many ascus, and it is a hook type of appendages. In Mycosphaera, you see there are you can say fringe type, or it's a fork type here. Here is a bulbous base in Phyllectenia. Here many ascus are formed. Okay, these are the ascus. Okay, UCP ascus. So this is by this way you can easily identify. In tomato, this is the libellula. You see, here we differentiate the pathogen on the base of the shape of the spores. Okay, these are the spores in case of Blumeraria. These are the species of libellula. Even in oidium also causing the powdery mildew of tomato. where you see this is the idea formation of idea. So these are the various characteristics, and you can see in case of Blumeria, the chains of the conidia. Here you can see chains of the conidia clearly visible. These are the theric conidia. Okay, the mycelial, the conidia are forming in chain. You can easily you have understand and you have seen in laboratory so many times. As for the symptom is concerned, I have told you that the pathogen causing symptom almost every crop. Here you see there is a powdery growth in the mango, powdery mildew, the powdery mildew fungus, the powdery growth on the inflorescence. Here you see in shade trees there is a powdery mildew growth. Okay, here is the powdery mildew growth. In tomatoes see the powdery mildew growth. In chili you see there is the powdery mildew growth. Powdery mildew growth in maize, in oat. The powdery mildew growth in pea. Powdery mildew growth in strawberry, in rose, in soybean, everywhere you can see the powdery mildew growth. In case of in case of wheat, in case of your grapes, see the powdery mildew, see this is powdery mildew of bean, powdery mildew of okra, powdery mildew of papaya. See it reduces the market value. This is the problem of the powdery mildew. It will attack on the fruits and all. It market value has gone. So here is the powdery mildew of the citrus. Here is the powdery mildew of linseed in the wheat. So you can see the pathogen how devastating it is. It is causing almost every crop. So now there is some insect also associated with the powdery mildew disease, which also help for the movement of the pathogen, or you can say it act as a vector sometimes. Not the pathogen inoculum in their body inside, but through it act as a carrier. You see there are lots of aphids is there, then red spider mite is there, and this is another insect which causing disease. This is called woolly woolly aphid, okay, which is a very common, and it is confused with the powdery mildew symptoms. Don't confuse. This is not the powdery mildew. It's a infection causing the woolly aphid. Okay. Here also this is a mealybug. It's also giving the powdery mildew like substance, but the association of the insect you can easily identify and differentiate that this is the powdery mildew. In citrus also there is a very common of woolly aphids and the mealybug. In guava also you see there is a mealybug, but don't confuse with this, because this is somehow looking whitish growth of powdery, but it is not the powdery mildew disease. It is basically due to the insect. These insect act as a carrier of the powdery dust, because it is moving from one plant to another plant. At the same time, spores are adhered to their body, and that spore can easily transfer to healthy plants. Okay, so regarding symptoms, I already told you that the white powdery fungal growth develop both surface of the leaves, petiole, everywhere. Okay, these are basically the asexual spores called conidia, which is developed on the surface of the tissue or surface of the host. Sometimes these, if the infection is initial infection in the lower side, then you will find some yellow spots on the upper surface also. And these whitish color is slowly become dirty and covering much portion of the plants, and finally the plants become dry, and it is very common. You see, this is powdery growth on the leaf. It can grow on the stem also in cucurbits. You see, I am talking about the now cucurbits. So in cucurbits, you see, in the final stage, the plant become dry, and due to that dry, there is a sunlight, enough sunlight, the fruit becomes smaller in size, its quality is deteriorating, it become mature early and ripen early. That's why its taste also deteriorating. So this is the reason. And sometimes this area turns brown and senescent prematurely. That means it mature early due to the infections, and automatically the leaf fruits become smaller and market value is less. Okay, so in later season you will find the attachment of the Clistothecia, that is the sexual fruiting body of the pathogen, and it is near about 0.1 millimeter in diameter, and you can easily with the help of a lens you can easily you see the presence of that Clistothecium on the infected portion in the later season. These Clistothecium generally act as a primary source of inoculum because it can survive in dead plant materials where it present in fallen in the grounds and all. So you see in cucurbits, you see the plant pathogen also occur at the lower surface also, and if it is infect the lower surface, in upper surface sometimes you will see the yellow spots, but don't confuse with the downy mildew. Downy mildew is something different. Here you see this is the downy mildew. These are the downy mildew. It's restricted in between the veins and sometimes giving the angular type of shape. Here you see angular shapes developing, but powdery mildew is totally powdery like substances spread on the surface, and it giving very smaller type of, not this type of downy mildew clearly. Here you see the association of the Clistothecium, okay, in the later season. These are the all Clistothecium. It is in June in large views. So you can see these are the all Clistothecium which having which generally contain the ascus and ascospores. And due to the infection, you see the poor rind color of the pumpkin here. These are infected here, poor rind color. So this all you will fetch less price by this disease. Regarding the pathogen, it's very interesting in case of cucurbits, because earlier we know that the cucurbits, Ericipi is the main causal organism of the cucurbit powdery mildew, but that concept has been changed. Now Podospora xanthi is the main causal organism, which was early caused as a Spirotheca fuligima, Spirotheca fucosa. These are the many synonyms. This is changing day by day. Maybe in tomorrow again they change something. Earlier we know the Ericipi sicuricium, but this perfect stage Globinomyces sicuricium, in many books you will find that it is caused by Globinomyces sicuricium. These are actually telomorph of this Ericipi sicuricium. So the Podospora xanthi is the main cause and Ericipi sicuricium is the secondary now. You can see a couple of informations. Podospora xanthi, previously known as Spirotheca fuligina and Ericipi sicuricium, these are the two important fungi related with the cucurbits powdery mildew, not only Ericipi sicuricium, which was considered to be the primary causal organism throughout the world before 1958. Today, xanthi, Podospora xanthi is the main cause throughout the world. So there is a lot of difference between Podospora xanthi and Podospora Ericipi sicuricium. We have to know, it's very important. You can't find many books because this is very important. So I have collected some couple of informations from the journals and many books. So you go through this. Podospora xanthi is more aggressive than Echinococcus. This is the number one. Then Ericipi sicuricium may have a lower temperature affinity, whereas Podospora xanthi appears to progress during warmer month rapidly. Okay. Now the conidia or spore Ericipicula and Jaticilla are difficult to distinguish, and Clistothecia both are producing, but the main criteria used to presence of fibrous body in the conidia of Podospora xanthi. This fibrous bodies is the main criteria by which we can differentiate. It's very difficult otherwise, okay, because most characters are common. During recent survey, Ericipi sicuricium was found rarely and only at the start of the disease development. in the USA, of course, but in India also nowadays, it's very contradictory that whether Janthi is more or Irisipicicorism, but in India also now the Podospora Janthi is the main causal organism. So you see the difference, both are causing the powdery mildew on the surface of the leaf, upper surface of the leaf. Here also you see in case of Podospora, but here both are forming the conidia in chain. Here also you see the chain, here also you can see the chain, but this is the fibrosine body. Okay, this is the fibrosine body which is not present in Irisipes sicurosum. That is the main difference. Okay, and these are the formation of the Clistothecium, which contain many ascas. Here also containing many ascas, mycelial appendages. Okay, so I have given, I found a list where you can find out what is the difference between these two. You can go through these various points. Okay, here also, what is fibrosine body? The conidia of Podospora janthi contain conspicuous discoid or rod-shaped fibrosine body which were first observed by Bounds, and they can be clearly seen in the spores in if you made the 3% aqueous potassium hydroxide solution, and that technique developed by the Cable and Valentine. So this is a little bit information about the fibrosine body, not that much information, but you have to know, because the fibrosine body is the main differentiating characteristics of Podospora and Sicurosura. Next, about the pathogen, the germ tube of pathogen are simple and straight, Podospora janthi, and it has a, sometimes it may be a fork, and the mycelia of powdery mildew are completely external, I already told you, and it produces haustoria in the epidermal cell, and from that haustoria it procures the food material. You can see this is the haustoria inside the cell, and from here it can procure the food material. This is the enlarged view of the conidia, or you can say sometimes it's called arthrospore. You know, it's an arthrospore like this because it is spore from a small conidiophore developed in chain. Here is the formation of the germ tubes. So at the end time you will form the Clistothecia, which are tiny, peanut size, spherical, and initially it's white but later turn yellow brown and finally black, as I shown in this figure. Okay, so this is act as a primary source of inoculum, and it can survive in the residues of the plants. So this is a picture wise how the spore can germinate and developing the epithelium, then develop the penetration peg. In that penetration peg, it inside the host cell, then procuring the sugar molecules from the host cell, and finally that sugar is converted to the mannitol, and that mannitol is taken by the fungus for their growth and multiplication. You can go through these things. Okay, here everything I mentioned, because from 0 to 6 hours what is happening in 10 hours, in 14 hours, especially for the MSc students, it is very important, because step by step I have given everything. You can't find in many books, it is not mentioned, but for your information and for your knowledge, you can go through this. It's very interesting, very easy. So this is the systematic position of the fungus, belongs to the eukaryota, fungi. This is known very well. Podospora janthi is self-explanatory. Regarding the disease cycle, and I told you that disease cycle is more or less same for every pathogen, every powdery mildew pathogen. Only there will be changes in this. Here you show the different type of symptoms, and there will be difference in Clistothecium. Somewhere will be mycelial appendages is here it is showing the simple mycelial appendages, but here some will be bulbous base, some will be hook type, some will be many ascus, some will be one ascus. Here will be only single ascus. Okay, these are the difference. Otherwise, everything are same. The conidia are forming in chain. Here, these are the things. Here is the conidia, is the mycelial network. It's sending haustoria inside the cells. You see, there are the haustoria, and these are the cells. A conidia. This conidia easily detach from the air current or any means, and this conidia easily germinate and infecting the plants cell and again forming the conidia. This is happening within 6 to 7 days, and that's why it is very fast progressing. Okay, and later it develop the clistothecium on the surface, in the older surface, older leaves, and sometimes it become dry, and these clistothecium actually basically it contains the ascus and ascospore. The ascospore released, these ascospore germinate and infect the plant. These are the very simple, you can easily understand, excellent life cycle of this pathogen, you can easily understand. So for if it is a for you say you are going for the powdery mildew of P, you just change the symptoms here, and everything is okay. Same. If you go for the rose, you mix the rose, and more or less same. There is no difference. Okay, so disease cycle clistothecium, for this you can easily understand from that the clistothecia act as the primary source of inoculum, and because it has so many it infects so many hosts, including all the weed hosts nearby vicinity of the crop. So all the times, especially some of the plants, they are infected by the pathogens, and from there the airborne conidia also easily come and infect the plant, act as the primary source of inoculum, or the plant debris which contain the clistothecium, that may also act as a primary source of inoculum. So clistothecium develop on the leftover whatever remains of the plants in soil for a less period, but it can survive from here. When it got the appropriate environment, it germinate, develop the ascospores, and that ascospore easily infect the plant. So this is way. Now this primary source of inoculum, I told you that clistothecium and plant debris and the so many collateral host one where the infection are present through inward conidia. Favorable conditions for disease development are the, if you planting dense population, it will create problems, low light intensity problems, high morning relative humidity, 90%, more than 90%, and of course cool and dry weather. It is for our condition cool and dry weather, but morning relative humidity is little bit favor the disease development. Keep it in your mind. Regarding the management, powdery mildew management, actually it's little bit difficult because as I told you that it infect both the surface of the leaf everywhere. So while spray, it's very difficult to cover the entire surface of the infected area, because in cucurbit's leaf are little bit border, and if you are spraying from the top, it is very difficult to contact with the lower surface. So during that period, you have to be very particular for selection of the equipments, selection of the nozzles, that it must cover the entire surface of the crop canopy. Otherwise, the effect would not proper. So you have to be very careful about the machinery or spraying sprayer, whatever you are using, and the nozzles by which your mist, it should be mist, and it should be cover the entire crop canopy. That is very important. So there are many chemicals used by the powdery mildew, but majority of the countries, especially the Western countries, they are going for the cultural management effectively, because their selection of the varieties, resistant varieties, then avoidance principles. But that means timing of or showing of the crop schedule, the change, and sometimes the clean cultivations and using of bio products. That means use of neem oil extract and so many mahua karanji leaf extracts. These are the things bio products they are nowadays using. But in our country, still we are using the chemical, and since we have the less areas, less hectareage, so we are not that much dependent on the bioproduct. So we are doing the chemical treatments. So in chemical treatments, as you know, that sulfur containing product is the best for the controlling of the powdery mildew. Generally we are using 200 mesh sulfur compound. We are using 25 to 30 kg per hectare. But this sulfur dust is not, it's generally phytotoxic for the cucurbit plants. So where we can go for the keratin, okay, 0.1 to 0.5% keratin. We can use 0.2%. We can use calaxin. We can use tridomorph, denocarb. There are many chemicals are available. You can go through this literature. You will find in USA they are using potassium bicarbonate based products such as Kelly Green and Millstop. I don't know whether these products are available in our country or not, but we have to know that this is the product they are using in US and other countries, because while you study you have to be develop certain global perspective also at the same time. So powdery mildew in European countries, the powdery mildew on trees such as apple is controlled effectively with the spray of sterile inhibiting systemic fungicides. This is not using in our country, but they are using for the grapes and all. Powdery mildew, they are also using, they are also controlling by using the phosphate salt solution detergent and ultra fine oil. They are also using in the greenhouse. Blue photosynthetic polythene sheeting they are also using. So these are the new things you have to know, though these are very limited in our country, but those are cultivating the crops nowadays in the greenhouse, they can apply, they can use this. Regarding biocontrol control, there is a certain pathogen that is called amylomyces cuscles. Actually, this is a pathogen fungus which mechanism act as a hyperparasites. What is hyperparasites? That means it grows so fastly and it covers the growth of the pathogenic fungi and it suppresses the pathogenic fungi so that pathogen die. That is the simple. So these pathogen act as a biocontrolling agent. So this pathogen, these are the fruiting body, pycnidia are forming by this biocontrolling pathogen, but this is a very good pathogen used against many crops, especially in the greenhouse. So this is the only biochemical biocontrolling agent available for controlling of the powdery mildew with based on hyperparasite. So experimentally, powdery mildew cannot has also been obtained through spray with biocontrolling agent. It is not 100% giving control, and it is activity completed, because as I told you that it's difficult because it's a covering of this entire surface of the leaf with the help of biocontrol and control the each and everywhere with the fungus. It is difficult. That is way, though it's giving enough reduction in disease development. So several biopesticides approved for organic productions in USA, and which includes such as botanical oil, biocarbonates, hydrogen dioxides, lipopeptides. They are using oil-based product like eco erase, jojoba oil, zem, stylet oils, paraffinic oils, neem oil, this is available in our country. Sesame oil we can use. These are applied for the control. So these are the recent informations you have to be know. So this is all about your powdery mildew of cucurbits. I discussed, as I told you, that most of the powdery mildew symptom, mechanism of infections, disease cycle, then factor for predisposition of the disease, all are same except your management. So here management of pea powdery mildew, here you see more or less same keratin everything, but here couple of varieties: JP83, PM02, JP4, JRS14, Usha Panna, Arka Ajit, Hunch, Ajat P4. These are the resistant variety I have collected from many sources. These are the resistant variety of powdery mildew. Regarding the rose powdery mildew, you just change this photograph over here. Everything is same. There is a formation of the conadial chain, formation of the clistothecium, then the ascus, ascospores, everything is same. And you go for the resistance cultivar only. Ashini, Ambika, Angels, American Pride, Surovi, these are the resistant cultivars available in India against powdery mildew of rose. More or less symptoms, survival, then your control are same everywhere, the same chemicals. You go through the literatures, you will find easy to understand. It is very easy. If you are facing any problem, you can ask me 100 times, no problems. So thank you. This is all about. Students, have a nice day.